All right, welcome back, fellow scientists. So today we are going to prepare for a lab that we're going to be doing on Friday, where you guys are going to get to use the microscopes again, and you're going to look at onion root tips, the cells of onion root tips, whose DNA has been stained so that you can see different cells in different phases of the cell cycle. So help me out. Onions, are they prokaryotes or eukaryotes? Yep, good job, they're eukaryotes. And then with the eukaryotes, are they plants or animals? Remember, we're talking about onions, right? Okay, good, they're plants, okay? So then plant cells, think about plant cells. Are plant cells more roundish or are plant cells more squarish? Okay, good job, they're more squarish. So we're looking for square cells. That's what we're going to be seeing. Now, nucleus in a plant cell and in an animal cell are both round. Right, so find my mouse right here. Hopefully the mouse is, or the cursor is highlighted or whatnot on the video. But you can kind of see the faint outline of the cell right here, and then so here's another cell right here, okay, and then here's another cell right here. And sometimes there'll be cells overlapping other cells, so it'll be a little bit different, but you can fight through the difficulty and you can get that. So if these boxes are cells, then these round things right here must be the nucleus. Okay, so all of these things are nucleuses, and the slides that we're going to be looking at have been stained so that the DNA is highlighted. So all of these dark lines or blotches or squiggles or whatnot, all of those things are DNA. Right, so let's start with the phase that you'll see most often, interphase, because that's the longest phase of the cell cycle. Right, and we talked about how there's different levels of condensing of DNA, right, from just the double helix, right, the genes that are being used, the DNA that's being used, all the way down into the most compact, most condensed chromosomes. Okay, so here, right here, we would see a nucleus of a cell in interphase, right, and we can't really tell if it's in G1 or S or G2 because again, we don't see any chromosomes. It's just kind of all spread out and all strung out, and it's not very, very tightly compact into the box, into the moving truck, right? Can you guys tell me what these little dots within this nucleus, what these little dots are? Yep, so those are the nucleoluses, right? I think on our diagram, maybe there was only one nucleolus for the nucleus, right? But you can see a lot of these have two nucleoluses here. This is a nucleus right here that just has one, right? So you'll see one, you'll see two, but that's what you're looking at here. Okay, so after interphase, then the next phase is prophase. Okay, so this is labeled right here so we can see a good diagram of prophase or a good nucleus in prophase right here. So the DNA is no longer just kind of spread out and all everywhere, but we can see it's kind of scrunching, it's kind of condensing down into chromosomes. We can see the little stringy things, right? So prophase and then metaphase, right? So here, so this is the middle of the cell where all of the centromeres are lining up, right? And so then we can see these little stringy things, these little fingers, right? Let's see, how does it? Yeah, kind of like that. These little fingers, right, are chromosomes, and all the centromeres are kind of lined up, right? So that's metaphase. Anaphase then is the chromosomes being pulled apart, so we can see, right, the spindle fibers. We can't see the spindle fibers, but we know that they're there, are attached to the cell wall, and they're pulling the chromosomes to one side of the cell or the other. So prophase, metaphase, anaphase, and then let's see, do we have a good example of telophase in here? We don't. So we need to go to the next slide to look at telophase. So this is a little bit more zoomed out, and this is kind of more like what you're going to see in lab on Friday, right? So, and these aren't labeled, which is intentional. So let's see if I can, there we go, zoom in, right, a little bit. So you'll see most of these, like this whole column right here, all in interphase, right? I would say that one's in interphase. That one we can start to see some tiny little dots right in there, so that one might be a good prophase. Here this one looks kind of squiggly. Here this one looks kind of squiggly, so that might be a good example of prophase. Here that one might be a good example of prophase. Right here we can see some definite like stringy thingies or white space. Maybe what you're looking for is white space in the nucleus. Right, so that's prophase. Metaphase. Okay, would be right here. So those are the chromosomes lined up along the middle. Anaphase, is there a good? Okay, so here's a metaphase right here, chromosomes lined up along the middle. Here's a metaphase right here, lined up along the middle. Metaphase right here, lined up along the middle. Anaphase is the shortest one, and so that's pretty hard to find. So we have a lot of late anaphases, or maybe like directly into telophases here. I would say this one right here, we can kind of see like the chromosomes. They're still we can still see a little bit. So they're just getting done pulling apart. So that's late anaphase. That's a good anaphase right there. I mean, it's not perfect, but you can kind of see it. So, and then telophase, we have a lot of telophase. Okay, so here we have a good telophase where now the nucleus, the nuclear envelope is starting to reform, and we can see the DNA. We can't see any chromosomes, any stringy things anymore, but it's kind of becoming one homogeneous blob again, right? Here's a good telophase right here. And then after telophase, then what happens? Yep, that's right, cytokinesis. So cytokinesis in plant cells, we would see that cell plate that starts to form. So if you look really, really closely, let's see if we can zoom in even a little bit more. Right, really, really closely right here. So here's the nucleus, and then here's the cell plate that's starting to form right here. And then I think there was another example. Oh yeah, here. So here's a nucleus, and then here's a cell plate that's starting to form right there. So that would be a good example of cytokinesis. Okay, so now that I've kind of taught you what you're looking for, right, what I'd like you to do is I'd like you to go and take the Microsoft Forms quiz. And on there, I kind of highlight a cell, just like this is, and I ask you what phase it's in. So I'd like you to take a guess. Okay, your choices are interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Right, so 6 choices, I guess. So those are your choices. And then I also ask you why you think it's in that phase. So talk about what the chromosomes are doing. So you can say either we can, like for interphase, you can't see the chromosomes, right? There you see the nucleolus, right? Something like that. For prophase, you can see the chromosomes are starting to condense. We see little dots, we see little spaghetti noodles, we see white space in the nucleus, right? Something like that. Metaphase, obviously chromosomes are lined up along the middle. Okay, anaphase, chromosomes are moving to opposite sides of the cell. Telophase, chromosomes are separate in their nucleuses and they're starting to unravel, and the nuclear envelope is starting to form. Right, and then cytokinesis is we can see a cell plate in between the two newly formed nucleuses. So good luck with that. We'll talk about this more on Friday. You guys probably have questions. Email me with questions, or we can talk about it before you do the lab on Friday. So this has been Mr. Leo. Thanks so much for watching, and I will see you on the flip side.